Okay, so we should continue, but first Professor Schulten wants to make a comment regarding this echo phenomena.
Relaxation processes, meaning molecular statistical mechanics. It's a wonderful example in linear response theory. And so it really teaches you a lot. It's just like that famous harmonic oscillator problem that we are encountering when we are teaching or when we are taught physics and chemistry. We learn so much from that bloody oscillator. What is it good for? Yes, in a way, yes, it's everywhere. But on the other side, it's really also a great teacher for training. And so this workshop is about introducing you to the concepts and the doing of molecular dynamics in this regard as we describe it. So,
it clearly tells us that the coherent motion in the proteins is of order of picosecond timescale. So whenever these kind of motion, coherent motion is relevant, you have to keep in mind that the time scale you're talking about is picosecond. All right, so let's finish up these applications of statistical mechanics of dynamics of proteins with another interesting example, namely the simulated cooling of proteins. We want to see how can we transfer heat through a protein. What is the thermal coefficient, the thermal conductivity for a protein, and whether it can be calculated using molecular dynamic simulations. So, what is really interesting about proteins is that they function in a very narrow range of temperature, so-called physiological temperature. If the temperature changes too much, it increases or decreases, then the protein loses its function. And therefore, it's interesting. We can learn about these properties of temperature-dependent properties of proteins by heating it up or cooling it down, and see how fast energy is dissipated or is transferred between different degrees of freedom. So the question which we really want to address is whether we can use molecular dynamic simulation to study the heating or the cooling process of a protein, and if yes, how could we do that? And also, what can we learn out of this? I mean, why is this useful? I mean, what are the properties which we can learn from here? So here is again ubiquitin, assuming that it is at equilibrated temperature T0. It's an equilibrium temperature T0. And how would you cool this protein? I mean, you should use a coolant. Experimentally, this is what people do. They would put the protein or the probe inside a coolant, which has a temperature T1, and then monitor how the temperature of a protein, with having some sensors here, would determine how the temperature of a system changes in time. And clearly, this is a non-equilibrium process. because it is produced by a temperature gradient. Whenever you have a temperature gradient, you have a macroscopic current of energy or mass. In this case, we are talking about heat. So it's energy transport from the protein, which is at higher temperature, towards the coolant, which is at a lower temperature. So in order to do such a calculation, in order to investigate the response of a protein to a temperature gradient, this is actually, it's a quite general way of studying condensed systems. Namely, you apply an external perturbation and you see what is the response of a system to that perturbation. If you are close to equilibrium, I mean, if a perturbation is small, then you can use the so-called linear response theory, which I already mentioned to you in my first lecture, that it is related to through fluctuation dissipation theorem. You can actually relate it to equilibrium properties of a system through correlation function. So the response function can be expressed in terms of properly constructed autocorrelation or correlation functions. Now, what we are going to do is the following. First, we are going to assume two approaches, a mesoscopic approach and an atomistic, an all-atom approach, or a microscopic approach. In a mesoscopic scale, on a mesoscopic scale, when we study a globular protein, we can regard this as a continuum medium. Continuum medium, it means that you're actually coarse graining your protein. You are dividing in small volumes, which still contains a sufficiently large number of atoms, say about 100 atoms. Of course, here we have only 1,000 atoms, 1,200 atoms, so we have to be careful and not to be too generous in partitioning the system, because otherwise you will have too few atoms in one little volume element, and therefore the mesoscopic description, it is questionable. But we will see that actually we can characterize quite well the heat conduction or the change of a temperature in a protein by using the continuum equation for the heat transfer. Namely, this is a result which is obtained in linear response. The time, the rate of change at the given point, the rate of change of temperature in the system is proportional to the second spatial derivative, or the Laplacian of a temperature, times a constant, and this constant, which is a kinetic coefficient, is called the thermal diffusivity, or thermal, or the heat diffusion coefficient. Here is actually what I mean by an atomic or microscopic and a mesoscopic description. In a mesoscopic description, in an atomic or in a microscopic description, you are taking into account each individual atom. This is what molecular dynamics simulation allows us to do. Each atom is treated individually, the relevant length scales of 0.1 angstroms or less than an angstrom, and the relevant time scale is about a femtosecond. Now, if we go to a mesoscopic description, now here I'm cheating a little bit in a sense that I'm representing the same protein but with the surface representation. So, but what I want to symbolize here is that I don't really care about each individual atom. I assume that I have really a continuum. I have a highly irregular form body which can be treated as a continuum medium. What we do, we coarse grain this. I mean, I consider that this is a volume element here, which already contains a large number of atoms here, and I'm averaging everything in this region. And this is what I call a point, or an infinitesimal volume. The length scale here is usually bigger than 10 angstrom. It's a nanometer scale. Even this one, it is, I mean, I expect quite big fluctuation to happen here and quite important derivation from a continuum description of a system. But as the size of a protein would increase, so if I would have a bigger protein, then probably the approximation would be even, I mean, this approach would be even more accurate. And also the time scales involved are clearly bigger than 1 picosecond instead of 1 femtosecond. So it's our 3 orders of magnitude difference here, and about 2 orders of magnitude difference for the length scale. So with the time scale, we are doing well, but the length scale, it may be a little bit too small, but this is what we have. So we want to probe whether the heat conduction equation really applies to ubiquitin or not. So let's go back to our heat conduction equation. So first I'm going to discuss this mesoscopic approach of determining the or studying the cooling process of ubiquitin, and then we are going to see how we can do this within NMD using molecular dynamic simulations. So again, the main quantity, so the kinetic coefficient which enters here is a thermal diffusion coefficient, D, which is related to the so-called thermal conductivity, which is characteristic to a material, to the density, and to the specific heat. So in order to calculate the thermal conductivity of a protein, which will be a number very similar to, or the same order of magnitude for all the proteins, all globular proteins, or most of them at least, you need to be able to calculate somehow the thermal diffusion coefficient, and then you also have to know the density. The mass density we already know, it's about 10 to the third kilogram per meter cubic, is exactly the same or similar to the water, the density of water. And then we have also the specific heat, which we have to calculate, and we know how to calculate it from the fluctuations of a total energy of a protein. An exercise or a problem which we are going to solve in one of the hands-on sessions. Now, we approximate our protein with a sphere of a radius r, which is about of 20 angstroms. And we are going to solve this equation, this partial differential equation. I mean, we don't really have to solve it because it is a well-known problem. These sort of problems have been solved 200 years ago. And we are going to use the solution for this particular boundary and initial condition. So the initial condition is that at t equals 0, the temperature inside the sphere is T0, and outside is the bath temperature, the temperature of a coolant. And this temperature will persist even at later times, because we assume that the coolant has such a huge heat capacity so that its temperature will not change at all during the cooling process of protein. So the protein releases heat until it gets into equilibrium with the medium, with the coolant. Now, before doing any messy mathematics, we can already do some back-of-the-envelope calculation, order of magnitude calculations. And you will see that by doing this sort of calculation, you can get your results on a at least correct up to an order of magnitude. So if you want more precise answers and get the right prefactors and everything, of course, you will have to solve the equation. So let's start from the heat transfer equation. And let's write it down in terms of finite differences. So here we have the rate of change of a temperature, which is delta T over tau 0. Who is delta T? Delta T is just the temperature difference between the initial temperature of a protein and the temperature of a bath. So delta T over tau 0, what's tau 0? Tau 0 is the characteristic heat transfer time. So it's the characteristic time during which the protein cools off. And this is an important characteristic which we want to calculate. And this is exactly the quantity what we need basically from molecular dynamic simulation. In molecular dynamic simulation, we can measure this. We can calculate this. Once you have that, we immediately can calculate the thermal diffusion coefficient, because delta T over tau 0 is just simply D times delta T over the distance square on which the temperature changes, which is of order of r. R is the radius of protein. If you work this out, you get that the heat diffusion coefficient is proportional to a distance square, so the size of a protein square divided by this crucial time, tau 0, which we don't know what it is. In order to get that number, which turns out to be about 10 picosecond right here, we have to use molecular dynamics simulations. So as I said, how do we get this time? From where did I pull out this number? Well, as you will see from MD simulations. Plugging in the numbers for R of order of nanometer, 10 to the minus 9 meter. As I said, tau 0 is of order of 10 to the minus 11 seconds. You will get for the diffusion, heat diffusion coefficient, a quantity which is 10 to the minus 3 centimeter squared. So it's a square is missing there over seconds. So as you can see, compared to the diffusion, the self-diffusion coefficient of a protein in water, this quantity is much bigger. There we get a number which was 10 to the minus 11. Here we have 10 to the minus 7. So the heat diffusion coefficient seems to be bigger. All right. Now, the solution to the heat equation. This is the solution. It is a very elegant way to, it can be solved very nicely using the method of separation of variables. And it turns out that the average of a temperature for the entire protein, so for the entire sphere, so I calculate the average temperature for the entire sphere, which means that you have to average over the volume of a sphere, and I'm going to get back to that through this plot here, is the initial value of this difference, which was simply T0 minus T bath, times an infinite series. But this infinite series, as you can see, it contains 1 over n squared at the denominator. So in a first approximation, you can just take the n equal 1 term. And for the n equal 1 term, you will get e to the minus pi squared t over tau 0. And the tau 0, which is r squared over d, appears again here. So you see why is this so important, this quantity? Because this is actually the time scale on which the temperature decays, the temperature difference decays to 0. So this is just another way of writing the cooling process, I mean the temperature change in a system, namely as a difference between, or a distance of a current temperature with respect to the final equilibrium temperature, which coincides with the bath temperature, T bath here. Now, how good is this approximation? Is it really legitimate to calculate the temperature of a protein, to talk about the temperature of a protein when we know that actually the temperature decreases along the radius of a spherical model of a protein? Indeed, at T equals 0, this is the temperature profile versus the distance from the center of a protein towards the coolant. So it's a very sharp change here. Inside the protein, the same temperature everywhere, and then you have a sudden drop to temperature, to the heat bath temperature. What is plotted here is, again, is the temperature difference. So that's why you go from 1, and this is also normalized, so that's why it goes from 1 to 0. Well, after a certain time, you see that the temperature profiles start changing. You see that even in the center of a protein, the temperature decreases. Then, along the radius of a protein, you see a more or less smooth decrease, especially after the first few femtoseconds. So, if you are happy with the idea that this curve here can be approximated with a constant temperature, so with a horizontal line, or something like this, so you have constant temperature which drops down to 0 outside the sphere, then this approximation is okay. If not, then basically what you will have to do, this is the approximation what we are going to do. Even in molecular dynamics, we are going to calculate the temperature of the entire protein. But you can do more than that. You can actually partition the protein in a way that you establish several spherical shells of different radius and just calculate the temperature according to that. You are not going to do that in the hands-on, because that would be more intense work. But you may want to do it yourself once you get home, or you know, just give it to one of your friends or your students, or to carry out this exercise. All right. This being done, we should go now and discuss the simulation of a cooling process using molecular dynamic simulation. So how do we proceed? Well, what we have to do, we have to change the temperature of a protein. There exist several methods to do that. And there are at least 3 of them. The one which we are going to use, and in fact is the most efficient one and the most realistic one, is the so-called stochastic boundary method, in which we actually are coupling the protein to a heat bath through Langevin dynamics. And I'm going to discuss that in the next slide. But I just want to say a few words also about the other 2 methods. Another method is the so-called velocity rescaling, or the method. This is very similar to what we discussed in the context of velocity of a temperature echo phenomena. So what you do, you know that the temperature, the kinetic temperature at a given time t can be calculated in terms of the kinetic energy of a system. Here ND represents the number of degrees of freedom. Of course, that is 3n minus 6. And if you want to change the temperature through velocity reassignment, what you have to do is to replace all the velocities by multiplying the current velocities with a square root of a desired temperature divided by the previous temperature. So if my temperature is T old and I want to go to T new, all what I have to do is to calculate, to multiply each velocity by this square root and restart the simulation. Now, this leads a very rapid cooling, because if, for example, I set T new equals 0, it means that I'm just quenching the velocities, and I immediately drop the temperature of a system by in 1 or 2, I mean a few femtoseconds to the half of a value of the initial temperature. Of course, I have to do it in a repeated ways. I have to do it several times in order to drain out the energy locked up in the potential energy. So it is not by setting the temperature to 0, of the velocities to 0, it doesn't mean that actually you are really getting down to, I mean, you reach a state in which the temperature of a system is 0. Of course, we don't want to do that. We don't want to change so abruptly the temperature, because that is clearly not compatible with the experimental situation in which the protein is put in contact with a thermostat, and a steady flow of heat from the protein to the coolant is the process which leads to the continuous change of temperature or decrease of temperature of a protein. Another way of changing, another way of doing this cooling is also related to reassignment of velocities, but in a different way, namely by random reassignment of the atomic velocities according to the Maxwell distribution. So in this case, you have more control, because you say, look, I'm at the T old and I want to decrease the temperature a little bit. So all what I have to do is to generate a new set of velocities according to the Maxwell-Boltzmann distribution and use those new velocities. Well, the problem there is that you lose any correlation between the previous velocity and the next time step velocity. So this is again unphysical. Whereas here you have highly correlation, so here the velocity autocorrelation function is not lost completely, but it is biased because you are using the same velocity scaled with the same quantity. So you are biasing somehow. But here you are completely destroying this autocorrelation. So none of these 2 methods are really realistic. The most realistic seems to be the stochastic boundary method, which I'm going to describe in a moment, right here. But before we do that, let me just mention to you that in an MD user guide, you have a chapter or a section which is called the temperature control methods, and you have several of those. You have the Langevin dynamic the temperature coupling method, and you have a temperature rescaling through velocity rescaling, and also the temperature reassignment through reassigning the velocities according to the Maxwell-Boltzmann distribution. All these sections here describe all the parameters which you have to set in order to perform the corresponding simulation. So here is the boundary, the stochastic boundary method. So it looks something like this. You have a protein. You surround it by water. Usually in this case, in the exercise which you are going to work on in one of the hands-on sessions, you will have a protein solvated in a spherical shell of water. It will be equilibrated, the system, at the temperature of 300 Kelvin. And then you will have to do the following. You will determine a boundary layer here, or a coolant layer of the order of 4 angstrom. It will be actually 4 angstrom, the width of this shell. The radius of a sphere, of the entire sphere, is, if I recall correctly, is 26 angstrom. So the inner part, the protein part, the radius of this inner part, it will be 22 angstroms. And now what you do is you assign different dynamics to the atoms which are located in the boundary layer or in the coolant layer, and different dynamics for the system, for the core or the interior part of the system. Notice that you will have even a few protein atoms which are part of the stochastic layer, so it's not only water molecules. And also there will be water molecules which creep into the inside or gets into the inside of a system which you build. And the reason for that is that the protein ubiquitin, you know, we want to really model like a sphere, but it's not a perfect sphere, so obviously you will not be able to separate in this simple way all the water molecules from the ubiquitin atoms. And you don't even want to do that because water molecules may even penetrate the system. Now, what is the dynamics, the equation of motion for the atoms in the boundary, in the stochastic boundary layer? Why it's called stochastic boundary layer? It's because the equation of motion will be stochastic. You have this Langevin force. So what you're doing is you're writing that the force which acts upon each atom, so mass times acceleration, will be equal to the force field, the force which comes from the force field or from the so-called native forces due to the interaction between the atoms described in the force field. And besides, you are also adding 3 more forces on acting upon each atom inside this boundary layer. One of these is a so-called harmonic restraint force. You want to make sure that the atoms will not leave this boundary region. So you really want to force them to stay there. Once they are assigned to be a boundary layer atom, you want to keep them there. But you don't want to tie them down. You want to give them a little bit of freedom to move, and that can be done by attaching them to a harmonic spring. As you will see, MD allows you to set actually the strength of this harmonic spring. You can make it stiffer, or you can make it lighter, so it's really up to you, depending what is the purpose of, or what are your goals in the simulation. You also have to add 2 more forces, namely the stochastic force, the Langevin force or thermal force, and the friction force. The friction force, Langevin force, or the thermal force, and the harmonic restraints, they are all related to the temperature through the fluctuation dissipation theorem and the equipartition theorem. So the equipartition theorem will actually determine the size of harmonic restraint force. I mean, this is how you choose the stiffness of a spring. Of course, you can put whatever value you want there, but you are advised that actually you should take a value which really corresponds to the temperature factor of a protein, which is nothing but the Debye-Weller factor. So you really want to reproduce to choose the right value for this and not to just to pick up an arbitrary value, though you may do that if this is what you want. Also, the friction force and the Langevin force and the thermal force we already discussed, and we saw how they are related to the temperature. Now, in a sense, you see that what you have here, this is nothing but a Brownian oscillator, on top of which you also have a force acted upon the atoms from the parts of the rest of the system, of the rest of a protein, namely due to the force field. So this is a fairly complicated equation, but of course it can be solved. This is not that difficult. NAMD already has built in all these features, so you don't have to worry about that. You just have to set up, you set the corresponding parameters. Inside the inner region, which describes actually the protein, which will change its temperature from initial 300 degrees to the 200 degrees. So I just forgot to mention to you that actually the temperature of the boundary layer will be set to 200 K. So this is how you're going to impose, to maintain this temperature through this coupling to this Langevin heat bath. Through the presence of these two forces, you will enforce that the temperature of this region will stay 200 K all the time on the average. And the harmonic restraint, the role of a harmonic restraint, again, is to keep the atoms there. So that is actually the purpose of these three forces. These two guys will keep the temperature of this region fixed, or on average fixed, of 200 K, and the harmonic restraint will keep the atom there and will prevent them moving into the inner region. The inner region, the atoms just follow the simple Newtonian dynamics, namely mass times acceleration is given by the force due to the all kind of interactions described in the charm force field or amber force field or whatever force field you are using. And now let me be more precise and refer to your hands-on, upcoming hands-on session, where you will going to simulate the cooling of ubiquitin. You are going to start, so this is actually the name of a directory or a folder which contains all the files you will need to complete this exercise. You will start from a pre-equilibrated system of ubiquitin at 300 K. The radius of a sphere in which ubiquitin was placed in is 26 angstrom. And your main task here is actually to set up the configuration files properly. Namely, you have to select the atoms which will be part of a boundary layer, because these will undergo different dynamics compared to the ones located in the inner side of a system. And for that, you are going to use VMD. You have to use VMD and this powerful feature of VMD of selecting atoms according to different criteria. So what you do, first you have to load your ubiquitin in the water sphere. You load both PSF and the binary coordinate files. I mean, VMD can do that. And now you are creating that coolant layer or boundary layer of 4 angstrom. How do you do that? Well, first you select all the atoms, both water and protein, so it's very simple. I mean, you are using TCL scripting to do that, or TCL commands to do that. Then we find the center of a system, the center of mass of a system. This is very simple. You have this command, measure center. You give a selection, and you also weight it with the mass. If you wouldn't put this weight pass, it will calculate actually the geometrical center of a system. But you really want to find the center of mass of a system, and therefore you really want to weight this average with respect to the mass of each atom. Once you determined the center of mass of a system, what you will have to do is to calculate the x, y, and z coordinate of a system center. And how do you do that? You loop through the I mean, this is actually a little bit a technical point because you need the x, y, and z coordinate of a center of mass. You could just simply use the get center get x, y, z command as well. But in a script you are given, because these are parts actually from a script are given, you are using actually another trick of producing the or getting the coordinates of a center of mass x, y, and z with this TCL command. Now is the crucial part. You are selecting the atoms in the outer layer. And how do you do this? How do you make this selection? So the selection you make is the following. You impose the condition that the distance of the atom with respect to the center of a circle, it must be smaller than 22 angstrom. And how do you do this? You square, I mean, the distance between the point of coordinates x, y, and z. So these are the coordinates of an atom, which you want to determine whether they are inside of a boundary layer or outside of a boundary layer. You calculate the distance between this point and the center of mass. So you have a square root of x minus, I mean, here it actually should be well, I wouldn't. So here, this is not square root, it's the square, that's sure. So you are squaring you're just writing down the radius of a sphere, right? So x minus x center of mass square plus the square of y minus y center of mass, z minus z center of mass, and this has to be smaller than or equal to the square of 22. So these will be the atoms which are inside. And if you are interested in those which are outside, you use this negation not, which tells you that all the atoms which are not inside are outside, basically. And now what you have to do, you have to set the beta value, you know, for each atom in a PDB file, you can associate two values, the beta and the Q, B and Q columns. And you can set these in order, set to 1 or 0, these values in order to make selections and pass it to NAMD in order to label them, whether these atoms will require special treatment or not. And this is exactly what you do here. You set all these atoms in the boundary layer, they set their beta value to 1. And you select then all the atoms now again, and you write them out in a PDB file. What will be with this PDB file different from the previous one? All what it will be different is that those atoms which are in the boundary layer will have a beta value equal to 1. So this is just a procedure of getting that. You already have a script there. You just run that script and you don't even have to bother with all these TCL commands, but I just wanted to go through that and to specify what they mean. And now let's see what are the special directives in the configuration files which actually specify what kind of dynamics you will have to use. First of all, you are going to use spherical boundary conditions. You will always see in your configuration files this weird blocks here with if 1 or if 0. When it's 1, it means that that particular feature is on. If it's 0, it means that it's off. So you will see lots of configuration file with IMD, interactive molecular dynamics. You don't need that, and therefore the 0 will be there. But there it may exist cases when you want to have IMD, and all what you have to do is you just flip the 0 to 1. So it's a convenient way of switching on and off different features of NAMD. So you are using spherical boundary condition. You have to specify the center of a sphere. You have to specify the radius of a sphere, which is 26 angstrom. You have to specify basically the strength of harmonic force, or the stiffness of a harmonic spring, which will keep the atoms to stay inside that particular spherical boundary of 26 angstrom. And here is just that you are really using a harmonic force kx squared divided by 2 is the potential energy. This is actually the default value. You shouldn't even, I mean, you don't necessarily have to write it down, but it's a good idea keep track of it. Anyway, I recommend that you go in the NAMD manual user guide and look up exactly the description of all these parameters. Then, you also have to specify the constrained atoms, which will be subject to the Langevin dynamics. And you are just specifying that the constraints are on. You give the PDB file in which the B column, which is specified here in this line, so this is the constant K column, it tells you that these atoms which have the value 1 or non-zero value in the B column will be subject to the Langevin dynamics method, or the stochastic dynamics, different from the rest of the system, which is subject just to Newtonian mechanics. And then you also have to specify what kind of coupling, temperature coupling you are having for this atom. So you are setting this on, that the coupling temperature is 200, and the temperature coupling file is given here. So as you can see, you have to specify twice this, boundary layer, making sure that you keep the temperature, you are coupling it to a heat bath of temperature 200 K, and at the same time, the atoms will stay in that particular shell for the rest of the simulation. Now here what I did, I just took out from the NAMD manual or the user guide this particular relevant part, namely section 632, which gives you a detailed description of how the temperature coupling parameters are set and what is their meaning. So I strongly urge you that you, when you run your simulation, you make sure that you understand each of these parameters, what is their meaning and why are they there. And the only way to do it, at least at the beginning, is to look up regularly the user guide and read the brief description, what is the default value, and what are the acceptable values. This can save you a lot of time when you're running your own simulation and it turns out to run in trouble and get some error messages. Now you have to run the simulation for about 10 picoseconds. That means that with the time step of 2 femtoseconds, sorry, this is femtosecond, you have to run it for 5,000 steps. This will take a while, but not that long. And then you have to extract the kinetic temperature, so the kinetic temperature T at every time from the log file or the output file, and then you can plot it directly using NAMD plot. And what you will get, and now I have a question here, are you sure that this is the right procedure of getting the T correctly? So you just have to double check this. Namely, you know that the temperature you're referring here, the kinetic temperature of an inner layer. The question is, the outer layer, is it calculated or is it taken into account or not when you do the calculation? How can you figure this out? Only if you know how NAMD works. And my understanding is that actually the outer layer is not included in this calculation. So because that is the heat bath. The heat bath, it stays at 200 K, so it makes no point to add it into the to the kinetic temperature. So when NAMD calculates for you the kinetic temperature, it calculates only the atoms which are subject to the usual Newton dynamics. If, however, you want to make absolutely sure, you can do the calculation yourself. You can extract the velocities from the DCD file and for the inner atoms and just simply calculate the corresponding kinetic temperature, and you will see that this is exactly what NAMD outputs for you. So NAMD does quite a lot of work for you, so it looks so simple using it and running it, and therefore it's a really great MD program, and also is very fast and scales very well. All right, so this is the black curve. What you get here is the temperature cooling curve. This is how the temperature of a protein changes, goes down from 300 K towards 200. Everything stops here at about 10 picosecond, at 10 picosecond. If you would run the simulation longer, you would see that you are getting a kind of critical slowing down as you go towards the 200K limit. So you probably want to speed up this cooling region here using different methods, like the velocity reassignment methods instead of stochastic boundary layer method. All what you have to do now is to fit your result to the theoretical prediction in order to see whether the mesoscopic type of description of a cooling of a protein, it is a good approach or is a good approximation or not for describing the cooling of a protein as a whole. And you will be instructed exactly how to do this fitting using XMGrace, a plotting program, which has also this nonlinear fit capability built in. Of course, you can use Mathematica, or you can use MATLAB or Maple, or your preferred mathematical software to do this fitting procedure. But regardless which one you use, it is very useful to rewrite your equation in dimensionless units. For example, as here, as you can see, since I don't know what is tau 0, I told you that tau 0 is of order of 1 picosecond. I didn't know that. Therefore, you don't know a priori. You really have to do the fitting in order to get that particular value. So the idea is to use a time scale, which is of order of 10 picoseconds. So I just use T0 as being 10 picosecond, and then X here is the fitting parameter, which I don't know. But I know that it is going to be of the order of unity. And that's important, because if you start messing here with numbers which are too big or too small, I mean 10 to the minus 10 or 10 to the plus 10 or something like that, you will run into problem. You get into serious round-off errors and numerical problems which will not allow you to get the right value. So it's, choosing the right dimensionless parameters is crucial. Also, you can always normalize this temperature difference, delta T average, with respect to its zero value, which is 100K. So 300 minus 200K is 100K. And then the only fitting parameter you are left with is X. And that is a fairly simple problem. As you can see, you get a very nice fit here. I actually use Mathematica to get this one, and it turns out to be 1.41, which means a thermal diffusion coefficient of 0.9 times 10 to the minus 3 centimeters squared over second, which is really very, very close to our order of magnitude estimate. You remember, we get 1. Now, this is really coincidence, because you cannot, with the order of magnitude estimates, you cannot really get the prefactor right, but we get the right value, 10 to the minus 3. Finally, let me conclude that this result can also be used to calculate then the thermal conductivity of ubiquitin. And to do that, you need, besides the thermal diffusion coefficient, you know the density, which we already have, and you also need the specific heat. And to calculate the specific heat, remember that the specific heat capacity at constant volume, or the molar specific heat at constant volume, is given by this expression. It's just determined by the mean square fluctuation of the energy. You know D, you know rho, you just calculate this. I'm not telling you what is the value, because this is one of your hands-on sessions. You are going to calculate that. But after you calculate that, just go back and figure out what is the thermal conductivity of ubiquitin, which should be actually a pretty good estimate for most of globular proteins of density of around the same value as the density of water. And I think it's time for me to finish up. Thank you very much, and we can discuss later.
Okay, so we can first ask a few questions during this lecture, and then we have our general question-answer period. So why don't we begin first with some questions regarding the thermal diffusion. So again, this is an exercise. We will, as we progress through the workshop, through the summer school, we will be getting what biologically relevant examples. So, who cares about heat diffusion of protein, but it's again color. One has sort of like a feeling for what kind of numbers they get. From this regard, it is an excellent first calculation to familiarize oneself with molecular simulations. You have a feeling for what kind of results should come out. It's doable in the finite amount of time. So you don't have to be worried that the whole summer school will be about heat diffusion coefficients and echoes and the friction constants. But it's an important conceptual starting point and a good practical starting point. Okay, so questions.
Yes?
When you set up the stochastic boundary layer,
Mm-hmm.
do you worry that some atoms, some molecules will be partially inside the internal layer and partially within the stochastic boundary layer?
Well, you don't let the atoms from the boundary layer to move in because you are restraining them with a harmonic spring. So if they would be tempted to move towards the interior, that restraining force will pull it back. So it's, they are all pinned down. They have limited motion, possibility for a limited motion, but they cannot really leave the heat bath region or the coolant region.
Right, but when you use statistical script to set up
Mm-hmm.
using the distance criteria,
Yes.
some of the atoms maybe just go across the boundary. And what does it do internally just to give it, to assign it to the stochastic boundary layer, or what did it do internally? I mean, in the VMD.
Oh, the VMD, well, you're just selecting the atoms, right, which are located in that layer, right, and you are labeling them. In the B column, you put 1 for those. All the rest of the atom have 0. So when you start running NMD, then NMD will know that these atoms have to obey the stochastic dynamic equation, so you act upon them with the Langevin forces and so forth, friction force, whereas the other atoms, they don't see those heat bath effects. I mean, they don't see they are not subject to the Langevin force and to the...
Right, but I guess my question is, suppose you have water, right?
Yes.
3 atoms, but one is...
And you take only one of those. Well, you can always do that by, if in your selection, you specify that the residues whose atom belong, for example, to the boundary layer, then all atoms from that particular thing to belong there. But let's take an atom. And how about if you just fix, you restrain the oxygen, for example, or on the proton. I mean, the water will not dissociate. So it will be like a water which is held back by a spring of one of its hydrogens, right? Or the oxygen. So for this particular problem, it doesn't really matter. But I understand you have to be very careful when you are cutting and you are separating atoms, that maybe some atom really work together with the residue in which it's part of, or the side chain it's parts of, and then you can always extend your selection criteria by specifying that all the atom, if this atom belongs to a side chain, then you either leave it alone or you take all atoms from the particular side chain. So you have all these flexibilities, all these features that VMD has in the selection of atoms. But here it doesn't really matter. As I said, you don't have to restrain all atoms in a water molecule. It's enough if you do it for one. Yes?
I think the thermal diffusion coefficient you calculate this
Mm-hmm.
way is the average thermal diffusion coefficient of the water and the protein, right? Because in the inner sphere, you have water molecules and protein molecules.
That's absolutely right. But you also have to realize that the total number of atoms in this system is fairly small. So whatever you get for this diffusion, diffusion coefficient it's a kinetic coefficient. It's a global quantity. So, but I agree. You are really calculating it specifically to this problem.
Yeah. So the problem is this thermal diffusion coefficient
Mm-hmm.
is dependent on the cutoff radius of the inner sphere.
Yeah, it's a very
So
interesting
if
thing.
you have a very big inner sphere, actually you're calculating the diffusion coefficient of water.
Okay, I agree what you're saying, but I'm telling you, if you do this repeated by making that layer not 4 angstrom, you make it 5 angstrom, or you make it, you make even a bigger sphere or whatever, you will still get the value, the order of magnitude of D, it will be 10 to the minus 3 centimeter sphere over second. So
I'm not talking about the outer layer. I'm talking about inner sphere.
in the sphere. Yeah, I mean, in a sphere, most of the atoms are still protein atoms. I mean, you can just count them. Go and count them. I mean, this is a good question. You can, when you do this exercise, you can do that and see how many water molecules do you have inside the inner layer and how many you have in the outer layer. And you will see that actually predominantly you have just protein atoms there. But of course, you can mix them, right?
Yeah, let's say if you carry out the simulated cooling on a protein which is not global, is it possible to set a boundary rather in other shape?
Yes.
Rather than sphere?
You can do that, but the idea is that then you will have to solve the heat transfer equation for that symmetry. And if you don't have any symmetry, then you have to do it numerically. But yeah, it's possible, yes. Can be, for example, cylindrical. For if you choose highly symmetric geometry, then you get nice results. Where's the next one?
young lady there, right?
Yes. Yes.
Okay.
Oh, to repeat the question, right? Okay.
I think it would be good if the questions would be actually addressed to the whole group rather than just to the speaker.
I also have a question. You are assuming that your protein can be described as a continuum for this approach of calculating the heat diffusion coefficient. How about using finite element methods? Because then you don't have any problems with the symmetry. Because you don't need a cylindrical or a spherical symmetry.
Absolutely, it can be done. Exactly, this was a previous question. It was, how about if you have not a globular protein, I mean, you're not really spherical, it maybe have a different weird shape, and all of them do have. Yes, I mean, it's just a different method how to solve the heat diffusion equation. But, for this purpose, here I think that it's better to start with the simplest possible thing. We are talking now about the prefactors, whether it's 0.9 or 1.2 or, it's not that important. But for research purposes, don't say and definitely don't quote that I said that the protein can be modeled as a sphere, because of course nobody will buy that. But, it's for zero order approximation, I think it's good, and especially for pedagogical purposes and learning the features of NAMD is suitable. But it's very good that actually you are thinking ahead and you're trying actually to get into more complicated and more realistic situation and how to address those. Yes, I agree. Use finite different element methods, you can solve the problem.
But I think it is really the fact that we have an analytical solution, the series expansion, which is actually the eigenvalue expansion of the heat diffusion operator for 3 dimensions, that makes us use a spherical case. There are two there.
Seems to me that in one of your slides, there is a picture of the protein. Most of the atoms of protein are in the internal layer, but some parts in the between the 2 layers.
Yes.
So I wonder if it is reasonable and feasible to include all the protein atoms in the internal layer. Otherwise, there will be some inconsistency for interactions for the atoms near and on the boundary.
Yes, again, I mean, the protein is not really perfectly spherical, especially this one. I mean, ubiquitin. I stick with ubiquitin because, it was the idea to have one protein throughout this, throughout summer school, because it helps you actually to focus more on the methodology and the methods you use to investigate the protein, and not to learn each and every time a new protein. It's an approximation. You're right. I mean, you should use not a sphere if you really want to, but you could actually consider just the protein itself, and you say that's the internal layer, and then the water outside is the coolant. But then it would be very difficult to solve the phenomenological heat diffusion equation, and then what's the purpose? I mean,
when you certainly don't get a paper published in Biophysics journal with a sphere or with any other shape or protein. It's not a hot research problem. It's just an exercise problem for you today.
It was 15 years ago maybe, but nowadays, you know, you have more interesting questions to ask. So, but yeah, I mean, the point is well taken. It's an approximation. It's a crude approximation.
But actually it is an intellectual, a very interesting problem. Some people actually work on heat diffusion in proteins because the question is how carefully do you have to describe all the degrees of freedom in a protein that participate in heat diffusion? You know, we have a classical simulation, and that means that every degree of freedom that is simulated has the heat capacity K. But you know from the famous Einstein formula that as you lower the temperature, the heat capacity of an harmonic degree of freedom goes to 0 at about the temperature where kT is equal to h bar omega. And now if you look at the protein, you find there are actually many modes where h bar omega is equal to or larger than K times 300 degree Kelvin. And so the question could be, and there are actually people working on this problem, do we get a proper heat capacity if we give every degree of freedom the same wrong heat capacity K in our classical approach? And the answer is yes, you get. It's a little bit of a problem it's a percolation problem. If you have, just imagine that you have traffic in Manhattan and you have a race of cars and they want to go from uptown Manhattan down to Wall Street. And now you, let's say bicyclists, and now you, let's say more bicycle, and now you generate roadblocks. And so like randomly you make roadblocks in Manhattan and you measure how quickly do they get there. And then you find that up to a certain number of roadblocks doesn't matter. You just imagine that the bicyclists know beforehand where the roadblocks are, and so they go their way until you hit for the first time the point that there are so many roadblocks that you don't get any more from the uptown to the downtown side. And that is a little bit like heat diffusion. All the heat flows through any degree of freedom there is, in this case through the classical degrees of freedom that have lower frequencies, and they can dissipate then the energy, in this case, from the protein to the outside until you have clipped too many passes that it doesn't work anymore. But there are so many classical degrees of freedom where h bar omega is smaller than K times 300 Kelvin, that actually the protein is very forgiving, and these quantum mechanical finesses don't really matter because there are other roads to get rid of the energy. But once you would lower the temperature of a protein, let's say to 10K, then you probably would see quantum effects in the heat capacity, in the heat diffusion. So in this regard, it's still a science problem, but otherwise not.
Yes? Well, but on this time scale of 10 picosecond, you won't see any major change in the conformation of a protein. Or you're talking about, I mean, deep unfolding and things like that?
No, not the unfolding. If you like do the simulation longer period of time, then you get the same value.
Well, what happens is you're looking to a non-equilibrium case here. You're looking how the temperature of a protein changes. I mean, that goes from 300K to 200K. After that, it will stay at 200K. I mean, you can run the simulation as long as you want, because if your integrator and force field and everything, they are right, its temperature will stay at 200. So you looked at a finite time effect there. I mean, you're looking at what happened at t equals 0, it has 300K, and then it goes down to 200. And it takes about 10 picoseconds to get there.
Okay. So before you leave, I have to make an announcement for the exercise today. Later, also we have a technical announcement. But I just would like to say now you can ask general questions. I asked my colleagues with whom we run together the summer school to actually come here into the room. So in case that there are some questions that Ioan and I cannot answer right now, they are available to also chime in. So if there's any question, but we will try to do this during the whole course of the school, any question you can ask them now. Yes, please.
In the lecture, you showed us slides from the user guide. Are we going to be provided the user guide? Or?
Well, that is available in PDF format online. So I think they. Yes?
Which user guide?
I mean the NAMD user guide, but the VMD user guide too exists. And
today,
yeah.
this afternoon, during the hands-on sessions, myself and the other lab people will have some CD-ROMs. So if you have a laptop and you want to get a copy of the new versions of VMD and NAMD that are being used for the summer school, you can get a copy of those. You can get a copy of the tutorial files, and you can get a copy of the user's guides.
So that means that some people asked if we can provide CDs with our programs, for example, with VMD for the Mac. Answer is yes, we will provide such a CD already today, and we might embellish that CD as we go on, and the CDs will be available in the workstation rooms, and you can copy them, and maybe we can also give you some before you go home. Now, in regard to the manual, to the user guide, you got a tutorial today. Tutorial is not a user guide. In the tutorial, we clearly indicate the URLs, the web addresses of the NAMD user guide, and you can find out then and get the user guide from there. Okay, so let's just go one by one. So please, and ask your question so loud that everybody can hear it.
A couple of brief questions. The first one is, will all the course materials for the SSO3 projects in Biocore in particular still be available after the summer school is over?
Yes, the answer is that we keep this summer school material on the web.
And the other question I had was, is there a publicly accessible computer facility on campus, like in the library or something, where we could just drop in and use it as a terminal?
There's not. Okay. If you have a laptop with wireless, there are various coffee shops around town that have internet, however.
Okay.
The closest we have is like you could all swarm the university, not university, city libraries. That's the closest we got.
We tried.
There is also a internet cafe that actually has computers to rent, which is on 6th Street next to the construction.
Are the rates pretty good?
I haven't used it, I haven't had to.
We tried, but we didn't succeed, unfortunately.
I've got a very consolidated protein, which I've got coupled to a heat bath, so I'm doing temperature coupling in a similar way described here. I'm coupling the whole thing to the same heat bath, both the solvent and the solute, and I find that, say, the solute is running hotter than the solvent. What could I do to address that problem? So could I couple both parts of the system to separate heat baths? Or?
Yeah, there were some problems with, I don't want to name names to make some people look bad, but there was some with the Groningen coupling to heat baths, where depending on the mass differences between the components that were coupled, you got exactly that kind of effect. So I think the technical answer is you should use one of the most recent versions of these programs coupling to heat baths to be sure that you avoid this problem. So your problem was, at least a few years ago, well known, and it has been addressed by the most recent coupling versions.
Am I right? Maybe Jim can also add to this. You have to speak up.
Okay. I was just asking if this was NAMD or a different program. So first question is, was the system fully equilibrated at this point?
Yeah, it's probably fair to say this isn't something I would say it can be. It's a phenomenon I'm aware of the calculated cells equilibrating ahead of time.
If you see it in NAMD, we would be very interested to address it. So if you could give us the files that produce that kind of output, we would be clearly love to look at it to see what was going on, particularly since that kind of behavior is known. Okay, then there was a question here. First, I thought there was a question here, then you had a question still? Okay, then you and you, okay. So it's you. Loud.
Okay. Since I'm just a newcomer in this field, I just want to ask a question in general. As I know, there are many force fields like amber, charm, and Gromax. So, in principle, is it important when I set up project, which system on which force field should I choose? And is it important I choose the right force field in order to get a right answer?
Yeah.
And what's the? I think each force field should have their own bias. What's the advantage, strong area, and the weak point for each different force field?
So this is too complex of a question to answer, but obviously, the simulations can only be as good as the force fields are. The force fields are not really perfect, absolutely not. There is a lot of discussion, and the various force fields are better for certain systems, like for DNA, some force fields like the GROMOS, and some the charm force field for lipids. There has been somewhat, since about a few years, less of a development in the force field area, because after the first excitement of developing molecular modeling, the group that developed the first program, the CHARM group, the Groningen group, the group at the University of California, San Francisco, EMBA, and also companies contributed to it. And I think there had been a little decay of that activity, but I see it coming again. I unfortunately see it also coming again that companies are actually going to sell better force fields. At the moment, for example, there's a lot of development of adding polarizability to the force field because it's felt that's a very important factor. And so I think I think we had some kind of renaissance of let's say, the Greek period of force fields when it all began. And then we had the Middle Ages. We at the moment, we are at the end of the Middle Ages, I hope. And now we hope getting into a renaissance phase where polarizability is added, where there's a convergence, for example, between the AMBER and CHARM force fields, because the developers, the untimely death of Peter Coleman and the CHARM and AMBER developer being very close together in San Diego, are very likely converging their force fields. Basically, they say already now they're not that different. The Groningen force field is a little bit of a force field that emphasizes more speed than accuracy to do large systems. Then there are companies out like Schrödinger and also some chemical companies that invest. We hope actually as a community to make a consortium that many people come together and ask the National Science Foundation, for example, to fund a force field activity for many groups so that it is being developed. Let's say in the United States, there will be maybe a force field. We found actually already a very nice name for that force field. We will be calling the Freedom Force Field. So that is sort of like the short answer. The longer would be now to go into detail. Which force field should you use for some particular applications? But the force field is, you know, a stumbling block. And I think the laughter I got by saying it's a complex question is totally warranted. Yeah, maybe I should say that our experience is that every time it matters, we actually have to do quantum chemistry calculations ourselves to really improve those parts of the force field where an enzymatic reaction is. Even if we have a water cluster that we think is important, we don't ask ourselves, should we now use tip 3 or tip 4p, but rather we use quantum chemistry because the electrons in a water molecule and several can actually couple to the molecules that are involved in the chemistry. They are like doing charge transfer, that some electron goes through the water molecule somewhere else. And that means you have to really describe it quantum chemically. And so that's also an important answer to your question.
I have a general question regarding molecular dynamics. So molecular dynamics is very useful to generate these statistical properties for the systems of interest. Then how do you make use of these statistical descriptions of properties to derive more deeper understanding of how this protein functions, or how do you make use of those properties, diffusion coefficient, heat diffusion coefficient, and specific heat? How those physical properties relate to the function?
Yeah, so I think this morning, if you ask this question in regard to what you heard this morning and yesterday, then the answer is simply we just calculate the simplest general properties that you can calculate for any protein in the PDB, because it's not related directly to the function, where you can find the values in the handbook of physics and chemistry, in order to just to get going, to show you that how you do calculations, how you abstract some real numbers out of the simulation output files that are provided. So I think they're not of direct importance, they are just training material. Now, in regard to the general question, what can you learn from molecular dynamics? That's of course a deep question that again, and please laugh now again, it's too complex, or very complex. Now, I think you have to go case by case. A wonderful source of information is in a recent account of chemical research, a special issue on, I think, 25 years of molecular dynamics. I think it came out either January last year. And so there you find some cases where molecular dynamics helped. It would also be very nice that maybe we did talk about it at some time, more systematically during this workshop. What are the moments of glory of molecular dynamics? And my general answer is that it's always an excellent method when it complements experiment. When it on one side makes contact with experiment, and on the other side extends what an experiment can tell you. For example, in atomic force microscopy, where we work and use a method, you measure certain properties, but just 2 or 3 numbers about the mechanical function of protein, like stretching a protein. You find out when I stretch it about 10 angstrom, I need a force of about 500 piconewton, and then it suddenly gives. Where does this resistance come from? So the experiment gives you the number, but a whole protein made of 5,000 atoms has a more complex story to tell than you stretch me by 10 angstrom and I resist up to 500 piconewton. You would like to relate that information to the actual structure, and so that is what molecular dynamics can do. So molecular dynamics can simulate the whole process, verify if there's some agreement with the observation, and then say, look, yes, it looks like, and now we can extend the picture, tell the full picture about the relationship between the architecture of the protein and the properties that are measured, and also the functional properties that are expected. Can suggest, for example, to the experimentalist, if you want to now probe the understanding, why don't you try the following mutants? And you say, make this mutant, this mutant, this mutant, they should affect these properties in the following way, and then one can have a give and take and check and recheck about our level of understanding, and finding, for example, the hot spot where the protein develops its mechanical force-bearing component. And so that is my still short answer. But it would be wonderful to have you know, a good-hearted but critical discussion about what can molecular dynamics really do for science. Is it more fantasy than fact? Is it really, you know, like it has many people who do it, but is it maybe pretty useless exercise? How much of what molecular dynamics publishes in the year 2003 will still be of interest in the year 2013, and things like this. I think it's a good discussion. Okay. Then the announcement I have to make is first, again, that the CD will be available and we will